Welcome to the PamGene data analysis training. My name is Dora, and I will walk you through all the steps. Here you can see the contents of the training. First, we have to design the experiments. The result of the experiments is a folder with images. There are images for every chip, every row in the chip, instrument cycle, and exposure time. These images need to be converted to numbers through a workflow called image analysis. Then these values have to go through some basic processing. Then we can analyze the kinases and the phosphosites in the data. The result of both of these analyses is information about the significance and the fold change of kinases and phosphosites, which can be plotted in different ways. Based on this, pathway analysis can be carried out to gain more insight into biology. Let's talk about how to design experiments. There are two basic experiment designs that you can do in a PS12 project. The first is when you have two model systems, for example, two cell lines, with the same test and control condition. This is called C1T1 C2T2 design. The second is when you have three test conditions and one control and only one model system. This is called the T1, T2, T3 design. You can see here that these are the replicates. Each replicate is on a different chip. In this experiment, there are three replicates. For experiments with cell lines, minimum 3 replicates are needed per condition. But if it is a primary cell culture or patient sample, then we need minimum 6 replicates per condition because of the higher variation. So essentially one more PS12 run. PamGene experiments are carried out using chips, which are small technical batches. It is important for correct data analysis that we design the experiment in a way that conditions are balanced over these technical batches. This is called a balanced design. The reason for this is that there can be variations caused by technical batches. For example, you can see here that even though the same samples were used, batch 2 has higher expression level. If the design is imbalanced, that is, the control and test conditions are in different batches, when we compare test to control, we can confound the real effect with the batch effect. Therefore, the design should always be balanced for conditions that we want to compare. And additionally, batch correction is also possible if needed. The next topic is image analysis. First, let's take a look at the file that we are going to upload to Tursin. This is a zip file that we get from the PamGene instrument. The name contains the 3 barcodes. Let's look into it. It has a folder called Image Results with the images. It has an array layout and 2 other files that we don't need for image analysis. You can leave them in or delete them. It's also important that since this is a PTK experiment, it has to contain the correct array layout with the correct chip type. The PTK chips start with 8641, and the array layout has to be named like this. The image results folder contains the images. Please don't rename the image results folder. This would lead to errors. Next, let's go to Tursin. This is my own team. You should see your name on the top. From now, please follow this training and do the steps after me in your own team. Here are the recent projects. Click on this plus to make a new project. Add a name and save it. I will go to the Teams tab now, to the PamGene Data Analysis Training Team. This is shared with you as well. This contains a project for the training of image analysis. I will work from here, but you guys please work in your own team in the project folder you have just created. When I make a project, first I always edit the README to add some relevant information. For example, the description of the project and which conditions we are comparing with which conditions, etc. It can come handy when later you have many projects and you have to find one of them. Then we have to upload the zip file that I showed you. So let's click on the upload file and I will drag it from here. And upload. The upload is done. The next is to make a new workflow. You can see that PamGene offers a few templates. First, we need image analysis templates. This is a PTK zip file, and there are two image analysis workflows, pre-wash and after-wash. So let's do pre-wash as an example. Let's rename the template. For example, template doesn't need to be in the name. But what can be in the name are the barcodes, or anything relevant, because you might have multiple workflows of the same type in the project. And you will want to distinguish them. The barcodes end with 030406. That is what I add to the name. Click on OK. Here is how the workflow looks like. First, we need to add the data in this data step. Here in the left top corner, you can see an icon that indicates that this is a data step. Let's run it and add the zip file that we uploaded earlier. It has turned green. Next, let's take a look at the images. Let's run the image overview step. The aim of this step is to see if everything is okay or if there is anything that has gone wrong. If there is an array broken or an artifact showing, this would be visible. This is done with an operator called the PS12 Image Overview Shiny operator. You can see the 3 chips, the 4 arrays on the chips, and you can filter for cycle. Now it's at 94. You can check any other cycle or exposure time. This operator is called a shiny operator because shiny operators are reactive operators, which mean that if there is user input like changing the cycles or exposure times, they will react to it. This takes a lot of computation power, so if you don't do anything with it, it will just stop working. But we can reset it and run it again. If everything is all right, then we are done with this step. But if you would like to document this and have a lab journal or similar, you can take a screenshot and put it there. Let's return to the workflow. We don't need to take a look at these 3 steps for now, so I will just run the regridding step and all the other steps before will automatically run. The aim of this step is to manually inspect every image and check if the gridding has been done correctly or if there are any small artifacts coming inside a spot. If the gridding is not correct, we have to replace the grid. We have to click on the grid button to check the next one and go through the images. When the signal is brighter, the exposure time is higher. Usually all grids are placed correctly. Misplaced grids are very rare, but I will show an example at the end. Here is how you can replace the grid if it is not correct. There are two ways. One is to replace the whole grid by clicking on the black part outside the grids, holding and dragging it. Place it in the correct position.
Part on the image and put it away so you can see that the whole grid is moving in this way. So let's say sometimes it happens that you find a grid like this and then you can put it back correctly. And then just click on the
Then
next.
just
If you
click on
go
the
back,
next. When you go
then
back,
you see
you
that
can
it was
see that the
saved.
grid was saved.
The other
The
way
other
is
is to
when we want
replace
to
only one spot
place
by
one
clicking
grid,
inside
then
the
you
spot.
click on in the middle of a grid and then replace.
You can click through all the grids. Please resist the temptation to be very fast, because it might happen that you don't notice something. By the way, this is again a shiny operator, so it expects interaction. So don't start it if you, let's say, want to take a coffee break during running. At the end, the grid button is turned gray. You can no longer click on it. Then push the run button. In the bottom right corner, you can see some running messages. It's running, please wait, etc. It is done now, the message is stopped, and the reset button has also appeared. Let's go back to the workflow and click on quantitation type step. While it is running, I will explain this enrichment file. The enrichment file annotates the sequences on the chip. The sequences have been blasted to human proteins. You can see the sequence match, the UniProt primary gene name and UniProt ID. Additionally, the sequence similarity, for which the threshold is 0.9, all matches above that or equal is in this file. You can also see that sequences have multiple matches, for example here. For example, ADD2 and ADD3 also match with a similarity of 1. Therefore, our data is much bigger than the number of sequences on your chip. These are all the potential proteins in your sample that can be phosphorylated. These are potential proteins because we don't have information whether they are actually in your sample, since we only know that these sequences can be phosphorylated on the chip. However, in certain cases, for example at pathway analysis, we can make this assumption. The sequence annotation is joined with the join step to the data through the ID column. At the end, you click on the export button. If everything goes well in the image analysis, this is all you have to do. One last thing, never forget to save your workflow. If the save button is orange, click on it. Otherwise, you will lose your progress if you accidentally close the whole page. Actually, the best is to save it during running as well, multiple times. Let's go back to the project. You can see that a new, exported data table has appeared. You can see in the name which workflow it is coming from. The names of exported files always begin with export, then dash, the workflow name, dash again, and finally the name of the step you exported it from. In our case, it was named as export. This is why it is important to always rename your workflows to know where the export files in the project folders are coming from. For PTK, there are two workflows to do, pre-wash and after-wash. For SDK, there is only one. The PTK after-wash template is only different in the filters. It works on the after-wash step, so it filters for cycle 94. If all goes well, you don't have to check anything other than the gridding, just run all the steps. Let's take a look at the exported file. There is a column called Instrument Unit, which has the name of your instrument. Then, there is barcode, row, exposure time, cycle, and ID, meaning that this row contains data of only one spot on the grid. Here we see the reference spot, but if I go to row 1000, we see one of the sequences, ZAP70. Next to it there is all the information from the enrichment file. Finally, the median signal minus background column contains the main output value of the image analysis. It represents the subtraction of the median background around a spot from the median signal of the spot. I will show you examples of workflow adjustments that you sometimes have to make in image analysis. This is an example of a small artifact, a tiny bright signal inside an empty spot. Let's place the spot away from the artifact. When the artifact is this small, usually this is not needed, since anyway the median signal and median background are used to create the output value. However, when the artifact is bigger, then this might be necessary to avoid false positives. Next, here is an example of wrongly placed grids. Let's run the regridding step. You manually have to replace the wrongly placed spots to the correct place, around the signal. Go through all the grids. In this example, luckily there is only one spot to replace. Go through all the grids and push the Run button when you are finished. The second example is about adjusting cycles. If the run in the machine has been stopped, the numbering of the cycle can change. What happens if we don't notice it? We get an error message. First, you can try to understand what it says. When you have already a little experience, you will notice that the first part of error messages are usually the same. It doesn't tell much. The real error is always at the end of the error message. However, in this case it is still not easy to figure out what it means. So, let's try to figure it out by inspecting the crosstab view. Let's reset this step and double-click. We can see here that something is not quite right, since there is nothing shown on the crosstab view. When we see this, our first thought should be, have we filtered for something which is not in the data? There is a filter, so let's remove it to see what happens. And the crosstab has now values. We can also see that there are only 54 cycles. So let's add the filter back, but now in a correct way. We have to drag and drop the cycle from the factors. Change the value to 54, since this is the afterwash, and also add a meaningful name for the filter. Now we can see that this is how the grid steps crosstab view should look like. Let's go back to the workflow and change the filters in the remaining steps. We don't see anything in the regridding, but now we get the missing factor warning message. This is because I didn't run the gather step before. Now I have run it, and we still don't see anything because of the wrong filter. So let's change the filter here as well. Again, the values have appeared because the filter has been set to a value that the data contains. Next, we have to change the filter in the select step because the quantitation type step didn't have a filter. Here we get the missing factor message again, but that's okay, since we didn't run the previous steps. Still, we can change the filters. It is useful to document in the project README that the experiment run has been stopped and the last cycle is 54 instead of 94. This will help us making the correct filters in the next data analysis workflow.